Okay, good morning, everyone, both in the room and online audience. Before we get started with this morning's presentation, Dominic and I just wanted to mention again that there'll be some changes to the schedule. And in particular, we're working to arrange things so that Thursday afternoon will be free for those of you who would like to go on some sort of other activity, sightseeing, et cetera. But then there will also be a discussion here for anybody who wants to stay in talk science. And then with that, I'm very pleased to be able to announce or to introduce Eduardo Sontag this morning, who comes to us from Northeastern University. And in a twist from yesterday's morning lecture, we'll be hearing about the mathematical model of the evolution of drug-induced resistance.
Thank you. I hope I don't trip on this. Okay. I don't need to tell this audience that robustness to anything, for instance, therapy, often incurs additional costs. So for example, these dandelions, this weed, compete with this other weed, which maybe requires more resources in the natural environment. But if we apply chemotherapy, AKA chemical warfare, we can get rid of them. But then we create a tumor microenvironment, so to speak, in which these guys take over. Maybe a picture that you'll remember a little bit more from this lecture. If we have a heavy armored cavalry soldier, that's a very expensive thing to maintain compared to infantry. So we don't need to allocate so many resources to the heavy armor and feeding the horse and all these kinds of things. And if we now apply target therapy, pun intended, we can get rid of the infantry, but now we have plenty of more room for feeding this horsemen, right? That then we cannot get rid of. So if nothing else, we remember these pictures from the lecture, okay? So one thing that we heard about several times, and we'll hear even more, and also in the previous iterations of this, the other workshops in this seminar series, there are things that we could try to do. We could try to do some sort of treatment holidays so that we can maybe kill the fragile individuals, but don't kill them all, so that we leave enough of the fragile individuals that they might outcompete because they need less resources, supposedly, than the resistant individuals. They might outcompete the resistant individuals, and then we can see whether we can keep iterating this treatment, but never killing all of the fragile individuals, right? So this has many advantages. One is about the competition between resistant and sensitive clones, so to speak, but also has advantages because, for example, the immune system may be damaged during treatment, and by giving these holidays, you have a chance to let the immune system reconstitute itself. So there's all kinds of advantages. And we know that, as we heard, for instance, in the workshop 2 weeks ago, there are even clinical trials right now going on for how to implement these kinds of things. So I never see anybody putting this together in one slide. I'm sure it's there somewhere, but let me just very briefly, in my view, think of what are the ways that people think of different kinds of treatments. So in red, I have what we might call a maximum tolerated dose, MTD, strategy, in which we give as much of the drug as we can without killing the patient and causing, kidney damage or, other kinds of problems. And then, wait maybe a month or a week and give another big treatment and so on. An alternative that is more along the lines of this treatment holidays, but maybe, or let's say of not killing everybody, not so much the treatment holiday by itself, is metronomic therapy here in blue. So metronomic therapy, stop like a metronome, right? You apply a periodic input, meaning drug, but at a lower level, okay? But you do it maybe more frequently. Another idea, and we saw, Josh gave this talk about metronomic therapy and the clinical trials for prostate cancer and so forth 2 weeks ago in here. Another idea is to use he was doing adaptive therapy, not metronomic, sorry. Adaptive therapy is therapy here I think in green, where we decide on some triggering events. For example, the triggering event may be that the cancer is over a certain level, as measured by PSA in the case of prostate cancer, or tumor mass or whatever. We apply treatment the moment that we cross an upper bound. We're getting a little worried. We apply treatment, and we stop treatment when we get to some sort of lower bound. And at that point, we decide we want to apply treatment because we don't want to attempt to kill all the fragile, all the sensitive cells. So we have some sort of feedback law, like a thermostat. If it's over a certain temperature, a thermostat has this gap, right? So if you're over a certain, say you want air conditioning, if you're over a certain temperature, you turn on the thermostat. And then if you're under a certain, and you stop when you get to another bottom, right? I don't want it to be the same, otherwise you get chattering, right? So a little bit of that idea. And more generally, in magenta, I wrote something a little more ideal, but I'll talk a little bit about that, which is full feedback, where we, in principle, could, in the adaptive therapy, what I showed was something where we applied the same amount, of drug in between triggering events, start or stop triggering events. In full feedback, what you do is you would apply a continuous amount of drug. And of course, we cannot do that right now, but there's no reason that we can't do it in the future through, either pills that you take very frequently with slow release, or continuous infusion pumps, like people starting to do with things like diabetes. So in principle, we could think of some sort of continuous treatment. That it's gonna happen. It's not right now, of course, but it's gonna happen. So let me start talking about the main things I want to talk about. And this was a slide from the Gillette and Gottman paper, I think it was, that Doran already showed the other day, same slide, about the possible mechanisms of resistance. So I'm not going to repeat it because he already talked about it for a few minutes. But I want to talk, the first part of this lecture, I want to talk about this issue of pre-existing resistance versus resistance that arises during treatment. And for the resistance that arises during treatment, I'm going to make a distinction that I want to talk about in the first part of the lecture. So prior to treatment, we might have a homogeneous population, or maybe we have some sort of mixed population. And as we apply treatment, think of the targeted therapy, right? Or the chemotherapy with the plants, okay? As we apply therapy, we destroy all the green guys, and then the red guys take over, right? Not good, because then we can't kill them, okay, with the therapy that is being used. And that step of going from here to here, you could argue it's sort of Darwinian selection under treatment. Our fitness has changed because now we have an environment that is not friendly to the sensitive guys, and they get killed and the other guys survive, survival of the fittest. What I want to focus on is the fact that this step of going from the mixed population to the resistant population can happen in at least two different ways, and I want to focus on those. One is pure Darwinian resistance. In other words, we're just selecting. The other one is Lamarckian, quote unquote, meaning that we not only do Darwinian selection, but we also generate variation. That's conceptually different from saying I'm doing Darwinian selection only. I'm generating variation, and I'll explain a little more what I mean by that, and then we do Darwinian selection. There's one more thing in this picture which I want to emphasize, and that's that often, especially with epigenetic changes. Now, with genetic changes, this is a little harder to think of because the probability of a backwards mutation, if it's 10 to the minus 9 or whatever, of course, then you get 10 to the minus 18 or whatever, that's really small. But if they're independent, right? But with epigenetic changes that can occur due to histone modifications, DNA methylation, or the activation of compensatory feedback loops, as happens, say, in the application of many inhibitors, RAS inhibitors, and so forth. Okay, those things are reversible. When we reverse things, we may go back to a mixed population, or maybe go up to a pure sensitive population. Now, this is a slide that was also shown by Doran. I've actually, he and I took the picture from Wikipedia, I believe. At least it is in Wikipedia. However, I edited the picture in ways that I explained to Doran the other day, not noticeable perhaps, but the old work of Luria and Delbruck, Nobel Prize in the, what is it, 56 or something like that, you said? 69, took a while, okay. They showed, and I'm not, and Doran already talked about this, so I was gonna go over this slide, but I won't unless you want me to, or I can do it later. But basically what they showed by doing this pretty interesting experiment was that resistance was pre-existing, did not arise during treatment. And in the end, the only thing you need to do is, well, not only thing, but basically what you need to do is you need to look at the Fano factor of the distribution. And if the Fano factor of the distribution is approximately 1, which corresponds to a binomial distribution in some limit giving of Poisson, then you can, as opposed to being much larger than 1, you actually can guarantee that what you have is, not guarantee, but if the model makes sense, you can say that what you had was pre-existing as opposed to resistance that arose in treatment. Now, in this case, we're not distinguishing between Lamarckian and Darwinian. I'm just saying it arises during treatment. Okay, so I'm not going to go through that because he did. And now, again, so the selection paradigm, as I said, is that you could have genetic mutations, you could have epigenetic mutations. They could be pre-existing, sometimes called standing variation. They could arise during treatment, sometimes called de novo variation. And both of those are Darwinian selection. As I said, I want to also focus here on the generational variation. So generational variation can occur in many ways during treatments. We know, of course, cytotoxic chemotherapies can increase mutation rates, and some examples of nitrogen masters, antimetabolites, and so forth, or they can induce various type of epigenetic changes. So I already said that before, right? So again, to maybe another picture to illustrate the idea, here we have, I'm pointing to for the people that are online, they wouldn't see that, but I think if I do like this, you see it better. So here I have a population which is heterogeneous. There are two kinds of cells, this particular type and this blue one, that actually are resistant to the drug. So as you apply therapy, you're just left with those, and now they can take over, right? Because there's no more competition, whether for vasculature or whatever resources, right? Oxygen, whatever. Now, there's also the fact that you might actually acquire resistance during treatment. Here, the cells were yellow, right? So those cells would have been sensitive, they would have been killed, but because of the use of the drug, they actually became resistant, and now they take over. So that would be the spontaneous, the acquired. So that would be the acquired situation, and that, as I said, could happen in at least two ways. One is that there's spontaneous changes, and then the drug selects the fitness change, so environment change, the fitness cost change, so we select for others, or it could be that it's induced. So all these things are just words, but and as I said before, I gave a short version of this last time. It is very hard to get your hands. It's like a very slippery notion. Like, what does it mean? And when I talk to oncologists, I talk to biologists, they usually tell me, what difference does it make? It happens during treatment. That's it. Too bad. I mean, why would we care about a difference? And I want to convince you that it actually does make a difference. But before that, the paper that actually got us started, I should have said it was in the first slide. This is joint work with Jana Geberts, who was here at the last workshop, with Jim Green, who was actually also in the slides, who's a former PhD student of Doran and was my postdoc for a few years, and we keep working together. Fantastic guy, as well as an undergraduate and the person that produced not this data, but the data I'm going to talk about later, Natasha Comandante-Lube. So the undergraduate, Samantha Prospieri, a brilliant undergraduate at Rutgers. Sorry, I moved from Rutgers to Northeastern, and I still keep saying Rutgers, Northeastern, okay? Which is actually in the northeast of the United States, not like Northwestern that is not in the West, okay? Or anyway, so this paper was really, really cool, okay? They're talking about this Lamarckian resistance. They had a particular in vitro study with leukemia cells that treated with vincristine and chemotherapy agent. And what I want to point out here is that after treating with a drug, this is a histogram showing the expression of MDR1, it's just one particular marker for resistance, okay? These pumps that Doran was telling us that pump drugs out of the cell, one particular type of those. And if you look at the, after various treatments, 60 hours later, various dosages, you get a probability distribution. This is a little bit like what Doran was showing us too. You get probability distributions of the different phenotypes. Okay, so this is the phenotype, right? That you get. This is not time, right? This is the amount of expression of this particular protein, this particular pumps. Okay, as marked through, I forget how they did the marking. Anyway, but what's really interesting in here is that you have this equilibrium probability distribution, right? That happens in the absence of the drug in magenta, right? Right there, right? That's an equilibrium probability distribution, okay? Now, if you take that equilibrium probability distribution and you take just the sensitive cells, which are the ones right here, the ones that don't express this pump that allows you to take the drug out, they actually, within about half a day, they repopulate to the wild type distribution. So you get some resistant and sensitive cells in this distribution being regenerated automatically. If you take the resistant guys, then after about, 17 days, you actually get back, if you look at the, what's in here, you get back to the, again, the purely wild type population. So what you get is you have this, there's of course, some sort of ergodic process or something going on. There's a Markov chain, when you really look at it, and there's some probability distribution for this Markov chain, and it goes to some sort of equilibrium, okay? I'm not going to do a probabilistic study, but you could do that, and people have sort of looked at this data probabilistically as well. But basically, there's some equilibrium probability distribution. So if you move out of that, you go back to this equilibrium probability distribution. So, but the only take-home message I want from that is not the probability, is the fact that resistance is reversible in this case, okay? And also that it's induced, even more for what I want to say, that it's induced by treatment, okay? So as you do treatment, as shown in this right-hand side, left-hand side, okay? Left for you, right? Yes. As you do treatment, you actually skew the distribution towards resistance, okay? Notice that there's a log scale at the bottom. So these are huge changes, okay? All right. There are many other examples of these kinds of induced resistance and even reversibility. I'm not going to go, I just have this in the slides. When you look later at the slides or the lecture online, you can look at them. We also describe them in our papers. So as I said, it's very difficult to actually get you, be precise about this. So let me do the following thing. I'm going to show how to mathematically think of the Lamarckian versus pure Darwinian models in a way that will show us why this could be of some interest from the point of view of therapy. There's a bunch of papers that deal with this that are listed in here. One is the NASCO Clinical Cancer Informatics. That's the first one we published things. There was an archive that we never published. If you want, I'll tell you later why. We weren't too happy with the results. There's a paper that I'll talk about a little bit about optimal control theory in Frontiers of Bioengineering and Biotech. And there's a paper under submission right now with new data that I'll tell you in a little while. So what is a model? The model is, so I believe in simple models, okay? Now, yes, you can have your model with 300 PDEs and 2,000 parameters, be my guest, have fun, okay? For me, I don't understand those things. I can see simulations, but I don't really understand what's going on. So for me, if I can have a model that is simple, that allows me to see interesting phenomena, I'm happy with that myself. Okay, just my preference. Okay, and you can even do some theory. Okay, so the model is going to be very, very simple. We're going to have two populations, sensitive and resistant cells. Sensitive and resistant cells. We're going to have a logistic growth. Later, actually, going to take out the logistic term for another in vitro experiment where we don't need it. We have growth rates. Okay, so logistic growth. V is S plus R, right? It's the total population. So it's just a standard logistic growth where we're competing for some sort of resource. I'm going to assume the growth rate of the resistant cells is less than the growth rate of the sensitive cells because you're paying a metabolic cost for expressing all these resistance mechanisms. There's going to be death rates for the sensitive guys, death rates for the resistant guys. I'm going to make the death rates of the resistant guys in the limit to be 0 for some theoretical analysis in a few minutes. I have a backward transition, which I'm also going to make 0 for some theory results. So that's given by this flow gamma. And I'm going to focus attention on these terms in here. So epsilon, U of t is a treatment, okay? Epsilon is something that doesn't depend on the treatment. So think of epsilon as the random or spontaneous changes that take you from sensitive to resistant. Gamma would be the ones getting you back, okay? They don't depend on the drug. It's random changes, epigenetic or genetic, I don't care. And they have nothing to do with the drug, and then the drug will select, right, by the differential rates of killing. So if there is no term with alpha in there, the red term is not there, then I have something where it's a pure Darwinian situation. I am sometimes changing from sensitive to resistant at random, maybe back, and then because of the competition given by the logistic term, they're competing with each other, right? So it's not clear already what's the best strategy, but now we'll make it more interesting and say, let's also suppose, and I make a log kill hypothesis right here, which I'll change later, but let's assume also that the drugs kills, affected, not kill, affected transition from sensitive to resistant at a rate which is given by this number alpha. Okay, so when alpha is non-zero, this is what I mean by induced by the drug. If there's no U, if U is 0, if there is no input, there's no drug, then that term is not there. But the term is proportional to the drug. In this simple example, you can make it more complicated, and I will. But okay, so is that so? Any questions at this point? And I'm going to make k equals 1 for simplicity. Any questions so far? Because we do have enough time. So, are there any questions now about the model? Sorry. What is log Q? Just that I have mass action kinetics. U times S. Yes.
How important would be the different growth rates that we put there to result the R and R?
Yeah, there will be, but I'll also give you a theory result that is very general, as you'll see. A theorem, okay, that says something will happen as long as the R sub R is less than the R. Okay, in other words, you want to have that the growth rate of the resistant cells is less than the growth rate of the sensitive. Is that okay, or there's a problem? Yeah. Yeah, and as a matter of fact, I'm going to give you another answer towards the end of the middle of this lecture. Okay, you'll see why. There's actually a very interesting thing that happens. But in this paradigm, we're assuming that you're paying a metabolic cost. The soldier with the armor is much more expensive to maintain, right? Or the weeds, the dandelions, need less than this other plant that was in the middle, right? Okay, that's the game I'm playing. I am totally aware of what you said, and we can discuss it later. So very quickly, I'm not going to go through these mathematical results, but you can prove that the parameters here are identifiable, and in particular from volumetric data, longitudinal volumetric data, and in particular, you can identify alpha. So at least in principle, mathematically, it's structurally identifiable. Of course, practical is a different question, but at least it's structure. You compute enough Lie derivatives, blah, blah, blah. You do the usual identifiability thing in mathematics. More interestingly, let's talk about how we can evaluate treatment. Remember, I make k equals 1, so the carrying capacity is 1 for simplicity. I'm gonna assume that gamma, the back transformation, is 0 for simplicity. So once you have resistance, you cannot become sensitive again. So clearly, no matter what we're going to do, what we're going to, and I'm going to, let the maximum possible value of S plus R be 1. So I'm thinking of fractions, if you like, right? But just normalize units so that the carrying capacity is 1, okay? So it is easy to see that no matter what we do, we're going to end up with everybody resistant, and they're not getting killed. So at the end of the day, no matter what therapy we use, what control you use, control in the sense of control theory, not in the sense of biology, okay? So what therapy you use is going to get us 0, 1 to non-sensitive full resistance, okay? But what we can do, we can still have an interesting mathematical problem here. We can ask, what is the best therapy to be able to be as long as possible below some level at which we consider this cancer to be bad? So we're going to have 2 levels, B detection and B critical. And I'm going to assume that no therapy is applied until I reach B detected. Okay, I didn't notice the cancer was there, whatever, at that, or PSA under a certain value, whatever. When I get to that value, I'm going to start applying therapy. Now, as I apply therapy, the populations at the beginning will, presumably go down, but eventually the resistant guys will take over and the population will go up. And I'm going to stop the experiment when I get to this critical point at which I give up on this therapy, right? Okay. So the objective mathematically will be to maximize the time to reach B critical, okay? And that's a well-defined mathematical problem. Is that okay? All right, so that's my mathematical problem. I want to let the patient be as long as possible before the cancer gets to this critical level. Okay, the first question I want to address is, like I said, some people will tell you, well, who cares? I mean, it's Lamarckian, Darwinian, et cetera, who cares? But I want to point out that, first of all, with the little simulation and then with the theorem, that in fact, whether alpha is 0 or not, in other words, where you have Lamarckian induction or not, makes a difference in principle. And here's a little simulation with some particular parameters. Sorry, simulation comes in a minute. What I'm going to do is I'm going to compare 2 therapies, one in which I give a constant dose and one in which I give a pulsed dose. Okay? And then the question is, which therapy is better when there's purely Darwinian or when there's Darwinian plus Lamarckian selection? And these are one set of results. These are all simulations, but this is one set of results. On the top, we have the case when alpha is equal to 0, pure Darwinian selection, okay? So the Darwinian selection happens through the carrying capacity and the competition, right? Okay, but the drug is not inducing the flow from sensitive to resistant that's happening independent of the drug. Okay, so in that case, constant therapy is better in the sense that we have a much longer time. I mean, these are the 2 strategies. There's a pulsed one that this is what happens with the tumor dynamics, the total volume, and we get to here the threshold is 0.9. And when we get to that threshold, the 0.1 was the detection threshold, we get just some numbers to the simulation, right? And when we get to 0.9, we have to stop, okay? And at that point, we don't can apply therapy, game is over. While if we apply a constant therapy in that case, we actually can do very, very well by, huge difference. This is mathematics. This is not really, no one is having anybody treating me based on this, right? But at least mathematically, it says there's some difference. And what's more interesting, perhaps, is in the context of the next thing, which is that in the case where alpha is not 0, and again, don't get hung up on the number 10 to the minus 2, and I didn't give you all the parameters, but some non-zero number. What you get is in that case, pulse therapy is better, okay? Because in the pulse therapy, you actually can last longer, not by a lot with these parameters, but you can last longer than if you gave the constant therapy that you have to stop much earlier. Okay, is that clear? So bottom line is that we notice that whether alpha is 0 or not makes a qualitative difference in the kind of therapy that you might want to apply. Again, it's just a simulation, okay? All right, but we tried many different parameters and you get sort of the same story. So yes, okay. Dominique, so, yeah.
Would that picture stay the same or change if you assume that the mutations that are induced by treatment occur during division?
You know, there's no cell division as such in this picture, right? So if you assume the population is asynchronized, then in the end, it's just changing the rate at which you're killing, right? So I'm not sure. We can talk offline. I'm not sure how I would model that
Right, I was thinking
in this model.
Rx times 1 minus x over k times limitation.
Yeah, let's talk about offline. Actually, the last part of the middle part of the lecture, I want to talk about something else that relates to that, where this is quiescent cell population, which is very related to that. Yes, Thomas, sure.
Did you keep track of the total dose which
Yeah.
is applied?
Right, right. So in this particular setup, you could argue the total dose was bigger in the blue case and the black case at the top, of course. But the point is, we're stopping the moment that we get to the critical point, right? So you could play the game where you have an area under the curve constraint, but it won't be a well-defined mathematical problem here, because if you stop too early, you never got to use all of it. So, but yes, that's an excellent question. No, in this case, it was constant. It was constant. So, so let me quickly formulate then to say, well, can we do this theoretically? So there's an optimal control problem, which basically says that when alpha is bigger than 0, when you have this induced resistance, in fact, you have more, you have to use more interesting strategies. This is an example of a numerical optimal control. Again, maximizing this time. Now the U of t is an arbitrary Lebesgue measurable function, blah, blah, blah. Apply control theory. And this is an example of what happens in one example where you get a bang of maximum therapy, holiday. Then you have this funny kind of therapy, which is increasing in time continuously, okay? And then you do a bang at the end, okay? And what's happening in here, it can be described in the following way. And these are now theory results. It's not for particular parameters, but it is assuming that the resistant cells grow slower than the sensitive cells. Let X be the trajectories for the vector field corresponding to no drug, Y being the vector field corresponding to maximal dose. I'm applying and now capital U, just for simplicity, is a time varying dose that allows you sliding along the critical line. Then the theorem is that the only trajectories that can be optimal are bang bangs, last bang, sliding, another bang. Okay, so that's a theorem. In the case of alpha equals 0, it's only one bang change. Okay, and we have not quite proved this mathematically completely, but the number of bang bangs that you have to use increases with alpha. Okay, there's some preliminary results about that. Okay, so it's what I said, that you need more pulsing, so to speak. Okay, so quick flavor for only the connoisseurs. There's some notations which obviously I don't have time to go over. You can actually prove that the only possible, let me just show the picture first. You can prove that the only possible singular trajectories occur along a particular line. Okay, sorry, on this one. Occur along a particular line. And what you can show is when you do it, and that takes a lot of work to get to that point. Okay, you have to look at the switching function. You have to look at the lead derivatives of the switching function. So the standard thing in differential geometric analysis of control problems that my colleague Hector Sussman, for instance, at Rutgers, had pioneered. But basically what you do is you show that, and then at the end, you show those trajectories cannot be optimal by introducing one form, which is sort of dual to which is equal to 1 on the uncontrolled vector field. The one for the lower values is equal to 0 on the upper one of the control vector field. And I should compute that one form explicitly. You can show that along any trajectory, the integral of that one form is just the difference between the initial and the final time. And then what you do is you actually show, by comparison with the bang trajectory, that this trajectory, the time that you're in here, is longer than the time that you have, shorter than the time that you have doing that. Okay, the velocities are different, so it's not clear from the picture, which means that one would actually take you less time, which is bad, because you want to maximize the time that you're under, this other limit, which I'm not showing. And that you do with a little, this little Stokes theorem argument and a little trivial differential geometry. So I'm not going to go through the details, but just to give you a flavor that this is a non-standard, this was Jim Green's detailed work to a great extent, and it's actually a highly non-standard optimal control problem. So I want to emphasize this was actually a pretty deep thing to go through this just to prove that theorem, because maximizing the time that you are in is what I call what is called a viability problem. Maximizing viability is not a standard optimal control question. All right, so some quick data fits to the data that we had in the paper that I showed you before, the Pisco paper from Shui Huang's group. We actually have some pretty good data, and actually we were able to, just from this data, looking at the model and the parameters that we got, we were able to predict some of the repopulation experiments. So that sort of says, oh, just from this data, I can find what is the population of resistant cells, what is the population of sensitive cells. I'm only measuring volume. So how can I do that? This model is, this is telling you how many you have of each. So you get all excited and you say, let's see if experimentally we can do something. So you could, so I worked a little bit with this group from Amy Brock, and in particular, Caitlin Johnson, University of Texas. They did this beautiful experiment with DNA barcoding. I'm not going to go through the details. A lot of machine learning that goes in to decide what you get, and I can explain it offline, not right now. But the bottom line is, you get some fits. You do this DNA barcoding experiment, and you predict. You actually, in some sense, you decide what is the total amount of sensitive and resistant cells that you have. Unfortunately, they only have 3 points. It's who was talking about many. They only have 3 points: intermediate point, the middle point, because they have to do qPCR at one. It's expensive, so at the beginning they do it at the end, and then they do one in the middle. So unfortunately, there's not enough data, but it failed miserably. So unfortunately, our model did not work. But I should say that maybe the fitting could be done better. So I've been meaning to reanalyze the data, and maybe some non-identifiability in some way, because they changed the model a little bit too, the way they did it. But the only thing I want to point out is you can ask, and this was the main point of that paper, what about if we now put together the DNA barcode in the expression data with the volumetric data? Can we get better fits, that fit both? And in fact, you do. And this is like the bottom. I mean, this is a little busy, but this bottom set of pictures shows what happens in that case. And the most interesting thing is when you use both the barcoded, when you fit parameters to fit both the qPCR data and the volumetric data, you actually get better predictions on the data that was not used for training. There was some data for other dosages that was not used for training. So it's sort of partial success, but unfortunately didn't work too well. But still, we tried. And the model is good, and I'm sure it's okay. But I think I would love to see these experiments repeated. Okay, and also there's a lot that went into the machine learning to decide what is considered resistant, what is considered sensitive, and I'm very happy to discuss later with you how it was done. And there's probably other ways one could reanalyze that data. So we had a simple model. We said the type of treatment matters depending on the level of, whether alpha is 0 or not, the level of induction. In one case, pulse treatments is better. In the other case, continuous treatments. But what we'd like to do is we'd like to sort of, we actually started collaborating with a different group and found much richer set of data. And that led us to the following new thing. This is the data that we use, working together with the first author, Natasha Comandante-Lu, the first author of this last paper in PLOS Computational Biology. And this actually was a melanoma cell line treated with bemurafenib and at various dosages. So what we did is, and I'm not going to go through obviously no time for the details of this, but this is the kind of data that they had. This is the response on the mean number of cells. Think of the volume V, the total number of cells. Okay, and you cannot, in that experiment, they didn't measure sensitive or resistant as such, although there's something I'm going to tell you in a minute that they did along those lines. Anyway, And what we notice is that no matter what treatment you apply, the three colors are three different dosages, okay? This is a logarithmic scale, okay? Log of negative 1.5, negative 0.5, and 0.5. At those three dosages, we notice that in the first 20 hours, there's no difference in the behavior, in the total volume, okay? The behaviors only arise later. There's some obvious resistance, the way that things sort of tend to, if you look at the yellow, it doesn't go to zero, it sort of stabilizes, okay? So that suggested that there has to be some sort of delay. So we modified our system. I don't have time to go through this. Let me just say in the end what the system is. We modified the system to allow for a little bit of a delay. So we assume that there's some delay on the drug action that produces the killing, but also on this term, we see the alpha term, the one that gives us the induced flux into from sensitive to resistant, and also again in the killing of the resistant cells. We set up these parameters gamma 1 and gamma 2 to 0.01, so roughly corresponding to 50% maximal effect at about at 72 hours. That was based on some literature, but then we fit the rest of the parameters, and they had 5 sets of doses, but we fitted 10 to the D of negative 1.5, negative 0.5, and 0.5, and keep the other 2 doses for validation. And we get an amazing fit. Okay, I actually love this. There's error bars from 4 replicates. We use an L1 loss function to fit. The fits are the solid lines. Okay, the fits are the solid lines, and it fits the data just beautifully. Okay, all right. Now we did we allowed the parameters to be different in each of them, but there's a huge number of data points. Okay, this is not poor data. This is not like that data with 9 samples or something. This is more than 3, Doran. Okay. This is a huge number of points. It's like every few minutes you have all the data. So we fit that really well. But for me, the interesting thing again is that we can, at least mathematically, predict what the sensitive and resistant populations are, right? So in the model, we have both the green and the black and the green and the dashed green lines corresponding to the sensitive resistant population. So we're guessing that those are sensitive and resistant populations. So that was a fit where we did talking about identifiability. So you can prove the thing is identifiable mathematically, but in practice also there's practical identifiability. Data is noisy, etc. We actually did obtain this amazing fact that the coefficient of variation when you take the sets of parameters that fit within 5% of the optimum, okay, just to take 5% as roughly a reasonable distance to the optimum of this L1 optimization objective, we get that the parameters sort of fit for these different colors at the different dosages. They fit very, very tightly. Okay, so really small coefficient of variation. That's telling us that it gives us a lot of confidence in this model. Okay, that the parameters are so identifiable. When we do the prediction on the doses that we had not used, we get pretty good predictions. This one, the one micromolar prediction wasn't as good, but we get the right behavior. Okay, we missed the peak a little bit, but other than that but actually I think there may be some problems with the data in that case. I'm a firm believer that if the theory doesn't match the data must be wrong. Okay, and I'm serious. Anyway. So but anyway. But in any case, we get the right orders of magnitude. We get everything right except for a little, a couple of hours difference, which I think is really good. Okay. Yes, I am going to be running out of time. Could we leave? Is that? Tell me the question. I might decide to answer it later.
Just validation going forward in time. Like if you were to restart the initial conditions, is that something you can do experimentally? Now that you know the fractions of cell types.
Well, so we're assuming. Okay. Yeah. Good question. So. Actually, that's an excellent question. I can't remember if in this case we fit the initial condition as well, or if we used the experimentally known initial condition. I have to remind myself. So there are 2 ways you can do this. Sorry, you don't have of course you don't have S and R, right? Right. But you could constrain S plus R to be equal to the observed volume, right? And I forget now if we did that or we fit both S and R independently on the data. But on the validation, I agree that, and I can't remember now. I'm sorry. I can't remember now. Yeah. Excellent question. I'm sorry. Can't remember now what we did in that validation step. But there's no cheating though. Okay, whatever we did consistently in everything. But I can't remember now because we did so much. We did so much in this project. We've been working for over a year or 2 years, and we tried all kinds of different things. And I forget what was the last one. Yeah, thank you so much for the question. Okay, very quickly, you could actually come out with a fine-grained model. So I said a little bit that they don't measure SNR separately, but in fact, that experimental data that you have has a single cell tracking. They do a little bit of what, again, we saw in Doran's talk, some of this lineage tracking. And we actually see that there's some populations that seem to be killed by the drug, others seem to be going longer at the single cell level, and some that seem to be quiescent. That somehow when you apply the drug, they just go into sleep. And this maybe has to do with Dominique's question about cell cycle. I don't think so, but it could be. In any case, we could write a quiescent model. We did, we fit the data. And what's interesting is that, in fact, we get the exact same fits, basically. So we have a huge number of parameters, not huge, many more parameters, but we get basically the exact same fits. So we concluded there's no point in looking at a more complicated model. And even more, some of these parameters now are hugely unidentifiable. I mean, look at that spread of these distributions, like the blue here, the yellow here, the blue in here. We get this huge distribution of parameters. So highly non-identifiable. We don't get any better fits. So why would you use this? Okay, Kathleen, we should talk about that because obviously that fits with the question that we're talking right before. Okay, so that's that. We also tried an optimal control formulation in this case. And basically what we find is that the optimal control actually works almost as well as a constant therapy in this case. Okay, not basically this is this is the volume at the end. Basically the both maximum tolerated dose and or even some sort of intermediate pulse doses don't do anywhere as well. You get a volume that is much bigger than you get with the optimal. Okay, but the optimal ended up being very close. So not terribly informative because it's just a simulation. We don't have the theory for this as yet. Okay, we didn't do theory. Okay, so the summary of this part, and I hope to go 5 more minutes, to add. I was told I could go a little bit into the part before the break, so coffee is not ready. So okay, so we talked a little bit about how whether it's with herbicides and chemical warfare, or it is with targeted therapy, we tend to select for the strong. Okay, what doesn't kill you makes you stronger. Okay, with some misappropriation of Nietzsche. We talked about emergence of variation could be spontaneous or not, and we talked about how we can distinguish how there are different therapy implications in principle for the mathematical model between having Darwinian or Darwinian plus Lamarckian plus Darwinian induction. And I talked also about this other data. So for this part, and as I said, I want to say one more thing that I added because of something that was said in the lectures yesterday, but that's my papers are there. This is Jana, Jim, Samantha, and Natasha. And very quickly, let me talk about a different aspect, which has to do with immunology. So we talk about resistance in terms of the competition between sensitive and resistant cells, right? And the fact that we make cells resistant, so the resistant cells then take over because we kill the sensitive cells. They're competing for whatever, oxygen, whatever. And now, once you took away the competition, the really bad guys take over, right? Okay, so you kill the bad guys, but not so bad that you could kill. And when you kill those bad guys, the really bad guys take over, okay? So that's one example. But there's a different way in which resistance can arise to therapy. And that's, there are many ways, of course, many ways. But one way is through what happens to the immune system. So what can happen is when the drug attacks the tumor, okay, it also kills certain components of the immune system. For example, if it's classical chemotherapy, going back to Dominique's question, particular cells in the immune system that divide fast can get killed, right? So you're shooting the cops, so to speak. Okay, all right. So you're damaging your own immunogenic components. So we got together with the group. This was work, by the way, together with Irina, who was here at the last workshop, okay? And my student, I'm Fong Tran, my postdoc, Mohammed Ali, but I got together for this work with David Waxman from Boston University, who's been carrying out a number of experiments about metronomic therapy and gave us a lot of really cool data. So, so that was a paper, and then we wrote a little model, and basically I'm going to show you equations, but really, for a lecture like this, doesn't mean anything because it goes fast. But basically, we have a drug compartment. There's a little bit of little pharmacokinetics that I will have in there. Very simple pharmacokinetics, but it's basically the drug in the microenvironment. That drug tends to inhibit immune cells, also tends to inhibit cancer cells. I'm going to have two components, X and Y, one of which is immunosuppressive, one is immunostimulatory. And the data that was used is data on, actually, this is in vivo experiments, okay, on mice. These are mice that have no adaptive immunity, okay, but it kept the non-adaptive immunity in there, okay? This is a model. I'm not going to, describe it, but basically, the immune components involve, in this case, involve natural killer cells, for example. There are no CD8 cells in this particular experiment, but and the immunostimulatory and immunosuppressive components include various kinds of cells that can, for example, could eliminate or increase the immune, antigen presentation cells and so forth, but as well as cytokines of various types, okay? I'll say something in a second. We use the data, as I said, from David's work. But here's some fits, which I want to emphasize, the parameters are the same, okay? So in all this data, so David gave us huge amount of data for different treatments with different dosages, different times between dosages that are applied periodically at different times, okay? Those are the dashed lines, the vertical dashed lines correspond to the times when the doses were being applied, okay? And we have here, and we fit with one single set of parameters. The only thing that changed was the amount of drug, okay? With one single set of parameters, only changing the amount of drug, we get this is the model, this is the experimental data, model, experimental data, model, experimental data, model, experimental data, and so forth for different kinds of treatments. You cannot see the captions, but CPA given every 6 days, 6 or 9 days in a metronomic schedule, or every 6 days in a metronomic schedule. This is the 6-9 is you do 6, apply drug weight 6, apply drug weight 9, apply drug weight 6, apply drug weight 9. So it's a little period 2 kind of thing, or purely periodic. And you see that we get just absolutely beautiful fits the whole thing, which is sort of nice. But then what is even more interesting, I think, is we have an immune component, right, in our model, okay, which we didn't use for the model. We just fit to the volumetric data. But now we said, okay, we have an immune component in our model. Can we see, and he actually measured immune components at a couple of days, okay? Again, low data, as Doran was saying. But at the days when the immune component was measured, we said how does our system predict that? And in fact, we found this is the top is the model, okay, and the predictions for the immune component. And if we look at the peak time, for example, in the 1 CPA, so done every day, metronomic therapy, and we look at various. His lab had actually done expression data. So we look at markers for various types of, even perforin in there, whatever, but various types of chemicals in the environment. They actually all peak about the same time and then go down. And the peak happens at the same time as our model was predicting. And the same thing in the, for instance, 2 CPA. In the 3 PCA, the peak occurred a little bit later around here, but it's not bad, right? I mean, we're pretty close. I mean, our model said the peak was there, but their actual peak was there, but it was pretty close. So I thought that was really cool that we can predict. Again, we had not used any of this immune data, and it can be predicted pretty well. We've done some optimal control, but that I'm not too happy with it. Tell you the truth, we did it. But then when we asked them to try experiments with this optimal control, they said they can't implement that in any way, even approximately, and it wasn't going to work. So they sort of gave up on that. So I'm hoping that, yeah, so I don't need the background. Okay, so anyway, I do not have I did in this particular slide, I don't have the people because I just added those slides because there was some question about immunotherapy the other day. So I figured I'll add those about this other work. So, but I should add to the co-authors, David Waxman and Mohammed Ali and Fong Tran and Irina Kareva, right? For the last part that I showed you, this slide I had done before that. But the papers are there and the reference is in whatever was Frontiers of Oncology or something, I forget. I get the reference before. So yeah, so that's it. Thank you for your patience.